Hello everybody. Today we will be discussing BHU PET 2014 MSc Plant Biotechnology question paper. To the first question, in diploids, number of linkage groups is equal to option 1 2n, 2 2n, 3 3n, 4 4n. Answer is 2n. Next one. To distinguish a double crossover product, we need at least option 1 2 genes on a linkage group, option 2 3 genes on a linkage group, option 3 3 genes on 2 linkage groups, 4 genes on a linkage group. Answer is option 2 3 genes on a linkage group. Next one. In rice, two dominant genes RC and RD are needed for red pericarp. If one or both of these are in recessive state, the pericarp is white. This is known as option 1 inhibitory, 2 supplementary, 3 duplicate, 4 complementary. Answer is option 4 complementary. Next one. Relationship between chromosomes related to descent in wheat are called option 1 homologous chromosome, 2 homologous chromosome, 3 heterologous chromosome, 4 orthologous chromosome. Answer is option 2 homologous chromosomes. A digenic test cross has a ratio of option 1 1:1:1:1, 2 1:1, 3 1:1:1, 4 1. Answer is option 1 1:1:1. Next one. Absence of one whole chromosome from a diploid is called option 1 trisomic, 2 tetrasomic, 3 monosomic, 4 isochromosome. Answer is option 3 monosomic. Next one. Which of the following organelles does not have RNA? Option 1 cytoplasm, 2 ribosome, 3 chromosome, 4 nucleolus. Answer is option 3 chromosome. Next one. Opening of flowers after pollination has occurred. Option 1 chasmogamy, 2 cleistogamy, 3 both 1 and 2, 4 homogamy. Answer is option 2 cleistogamy. Next one. Actual point of exchange of genetic material during crossing over. Option 1 chimera, 2 chiasma, 3 chi-square, 4 stigma. Answer is option 2 chiasma. Next one. Chromosomes are drawn to scale and the diagram is called option 1 ideogram, 2 ideogram, 4 karyogram, 3 karyogram, 4 karyotype. Answer is option 2 ideogram. It is IDIO, not IDEO. Next one. With higher annealing temperature, amplification becomes option 1 more specific, 2 less specific, 3 no effect, 4 average specific. Answer is option 1 more specific. Next one. The terminator sequence in genes prevent seed germination. Option 1 in second season, 2 immediate generation, 3 after 3 generation, 4 kills the seed after harvest. Answer is option 1 in second season. Next one. Independently assorted characters. Option 1 have linked genes, 2 segregate independently, 3 partially linked, 4 linkage is 20%. Answer is option 2 segregate independently. Next one. Addition or deletion of one or two bases to a DNA strand. Answer is option 1 frameshift mutation, 2 chromosome mutations, 3 insertions, 4 major mutations by addition. Answer is option 1 frameshift mutation. Next one. Theory of acquired character was put forth by option 1 De Vries, 2 Morgan, 3 Lamarck. Answer is option 3 Lamarck. Next one. The operon hypothesis was proposed by option 1 Watson and Crick, 2 Beadle and Tatum, 3 Jacob and Monod, and 4 Nierenberg and Corona. Answer is option 3 Jacob and Monod. Stroma and grana are parts of option 1 chloroplast, 2 nucleus, 3 mitochondria, 4 lysosomes. Answer is option 1 chloroplast. Next one. In a cell, the main sites of respiration are option 1 plastids, 2 ribosomes, 3 mitochondria, 4 lysosomes. Answer is option 3 mitochondria. Next one. In a cell, sites of protein synthesis are option 1 plastids, 2 mitochondria, 3 ribosomes, 4 lysosomes. Answer is option 3 ribosomes. Next one. Meiosis is also referred to as option 1 equational division, 2 homotypic division, 3 reduction division, 4 additional division. Answer is option 3 reduction division. Segregation occurs during option 1 mitosis, 2 endomitosis, 3 meiosis, 4 prophase. Answer is option 3 meiosis. Next one. The process of DNA synthesis from RNA is called option 1 reverse translation, 2 reverse transcription, 3 transcription, 4 reverse mutation. Answer is option 2 reverse transcription. Next one. In mitosis, DNA synthesis takes place during option 1 G1 stage, 2 S stage, 3 G2 stage, 4 all of the above. Answer is option 2 S stage. Next one. Equal division of cytoplasm during mitosis is called option 1 equational division, 2 karyokinesis, 3 cytokinesis, 4 horizontal division. Answer is option 3 cytokinesis. Next one. A chromosome with several centromeres is called option 1 acentric, 2 dicentric, 3 polycentric, 4 monocentric. Answer is option 3 polycentric. Next one. The movement of chromosomes at anaphase is associated with option 1 chromomere, 2 centromere, 3 telomere, synaptonumal complex. Answer is option 2 centromere. Okay. Inversion leads to alteration in option 1 composition of gene, 2 sequence of genes, 3 number of genes, 4 addition of bases. Answer is option 2 sequence of genes. Next one. Translocation homozygotes lead to alteration in option 1 gene number, 2 sequence of gene, 3 linkage map, 4 linkage group. Answer is option 2 sequence of genes. Next one. Monoploids are represented by option 1 X, 2 2X, 3 N, 4 2N. Answer is option 3 N. Next one. Allopolyploidy is referred to as hybrid polyploidy, simple polyploidy, segmental polyploidy, no ploidy. Answer is option 1 hybrid polyploidy. Next one. Laws of inheritance were discovered by Mendel in 1866 working with option 1 Drosophila, 2 maize, 3 garden pea, 4 neurospora. Answer is option 3 garden pea. Next one. A cross between two inbreds by reversing the order of male and female parent is called option 1 test cross, 2 back cross, 3 reciprocal cross, 4 dominant cross. Answer is option 3 reciprocal cross. Next one. Recent controversy of a GM crop in India is BT cotton. Next one. Highly cross-pollinated species among the following is option 1 maize. Next one. A vegetatively propagated crop is option 1 sugarcane. Option 2 sweet cane, 3 salt cane and 4 bitter cane. Answer is option 1. Scientific name of rice is option 1 Oryza sativa, 2 Oryza swastiva, 3 Oryza sativa and 4 Oryza sativa. Answer is option 1 Oryza sativa. Microsatellites are always found in bacterial genomes. 2. Always smaller than 50 base pair. 3. Used as DNA markers. 4. Repeated sequences. Answer is option 3 used as DNA markers. Transposons. Option 1 RNA sequences. 2. DNA sequences. 3. Only found in eukaryotes. 4. Contain no genes. Answer is option 2 DNA. Most genes are transcribed into option 1 tRNAs, 2 mRNAs, 3 ribosomal RNAs, 4 repeat elements. Answer is option 2 mRNAs. Transcriptomes, option 1 consist of RNA, 2 consist of proteins, 3 translated into proteins, 4 do not change. Answer is option 1 consist of RNA. PCR is used for option 1 digesting protein, 2 digesting DNA, 3 copying plasmids, 4 amplifying DNA. Answer is option 4 amplifying DNA. Transformation. Converts DNA into RNA, converts RNA into proteins, joins two DNA fragments, digest DNA into fragments. Answer is option 3 joins two DNA fragments. Next one, DNA polymerases. Option 1 join DNA fragments, 2 replicate RNA, 3 replicate DNA, 4 synthesize DNA in 5' 3' direction. Answer is option 3 replicate DNA. Genome markers must be option 1 must be repeat DNA sequences, 2 can be any unique DNA sequences, 3 are only used in genetic maps, 4 must occur as multiple alleles. Option 4 must occur as multiple alleles is the basic principle of PCR is performed with option 1 help of primers, 2 help of RNA, 3 restriction enzyme, 4 double stranded DNA. Answer is option 1 help of primers. GM is gene modified, 2 genetically modified, 3 gene multiplied, 4 genesis modified. Answer is option 2 genetically modified. Success of green revolution is due to option 1 development of dwarf varieties, funding from India, use of only fertilizer, use of only pesticides. Answer is option 1 development of dwarf varieties. Next one, plant DNA fingerprinting can be cost effectively carried out by option 1 RFLP, 2 AFLP, 3 RAPD, 4 SNP. Answer is option 1 RFLP. Next one, southern blotting is performed for the transfer of option 1 RNA, 2 DNA, 3 proteins, 4 carbohydrates. Answer is option 2 DNA. Law of purity of gametes is also called as option 1 law of segregation, 2 dominance law, 3 law of independent assortment, 4 law of assortation. Answer is option 1 law of segregation. Next one, micropropagation has direct relationship with option 1 tissue culture, 2 vermiculture, 3 silviculture, 4 sericulture. Answer is option 1 tissue culture. Two important components in tissue culture. Option 1 chlorophyll and cytoplasm, auxins and gibberellins, charcoal and alcohol, acetic acid and ascorbic acid. Answer is auxins and gibberellins. Option 2. Next one, gene gun is concerned with option 1 transformation, 2 electroporation, 3 conjugation, 4 callus formation. Answer is option 1, transformation. Next one. Agrobacteria were isolated in option 1, bacillus, 2, tumefacients, 3, crown galls, 4, TI plasmids. Answer is crown galls. Option 3. Ring spot virus is predominantly seen in option 1, papaya, 2, pomegranate, 3, potato, 4, peanut. Answer is option 1, papaya. Next one. Which country recently permitted the cultivation of BT rice? Option 1, Sri Lanka, 2, South Korea, 3, India, 4, China. In 2014, the question paper was set. So answer is China. Next one. First food product of biotechnology is option 1, cheese, 2, orange, 3, brinjal, 4, apple. Option 1, cheese. BT brinjal field trials in India were first carried out by option 1, Indian company, 2, American company, 3, German company, 4, Australian company. Answer is option 1, Indian. Main architects behind golden rice are Ingo Potricus and Peter Bayer, 2, MS Swaminathan and GS Khush, 3, GS Khush and Robert Ziegler, 4, Robert Ziegler and MS Swaminathan. Answer is option 1, Ingo Potricus and Peter Bayer. Next one. Which of the following enzyme was isolated from bacteria? Option 1, Taq polymerase, 2, DNTP polymerase, 3, exon polymerase, 4, Taq isomerase. Answer is option 1, Taq polymerase. Next one. The nucleotide sequence present in DNA sequence is option 1, ATPG, 2, ATGC, 3, AGCU, 4, AUTC. Answer is option 2, ATGC. Next one. Which of the following pair of sequences contain a single nucleotide polymorphism? That is SNP. Answer options are AAGGCTAA and AAGGCTAA. Next one. AAGGCTAA and AAGGCTGG. Next one. AAGGCTAA and ATGGCTAA. Next one. GGGG and TTTT. Answer is option 3. There will be in SNPs there will be change in only one nucleotide, and that is seen in option 3, where the second A in the first sequence becomes T in the second sequence. Next one. Standard gel electrophoresis of DNA separates DNA fragments based upon option 1, charge differences, sequence differences at one nucleotide, size differences, types of DNA. Answer is option 3. Next one. Agarose is a polysaccharide, 2, polydecaharide, 3, polyacrylamide, 4, polyamide. Answer is option 1, polysaccharide. AIDS virus can be detected by option 1, FCR, 2, PCCR, 3, PCR, 4, TCR. Answer is option 3, PCR. Next one. Callus is an undifferentiated mass of option 1, bacterial cell, 2, fungal cell, 3, algal cell, 4, plant cell. Answer is option 4, plant cell. Next one. The segment of gene that codes for protein is option 1, intron, 2, exon, 3, exene, 4, neon. Answer is option 2, exon. LCR refers to option 1, locus control region, 2, locus control receptor, 3, locus control regulator, 4, locus control reactor. Answer is option 1, locus control region. Next one. Plant protein that specifically bind carbohydrates is option 1, peptin, 2, lectin, 3, lactin, 4, mycosine. Answer is option 2, lectin. Next one. Mass in plant breeding refers to option 1 marker assisted selection, 2 marker assisted selection, 3 marker free selection, and 4 marker added selection. Answer is option 2 marker assisted selection. Next one. Proteins that oversees the correct binding of other protein is option 1 molecular neutrons, 2 molecular cutters, 3 molecular chaperones, 4 molecular scissors. Answer is option 3 molecular chaperones. Running several PCR reactions with different primers in the same tube is option 1 multiplex PCR, RT-PCR, 3 long PCR, 4 reverse PCR. Answer is option 1 multiplex PCR. An enzyme that uses single-stranded RNA as a template for making double-stranded DNA is called option 1 reverse transcriptase, 2 reverse transase, 3 reverse phosphatase, 4 reverse scriptase. Answer is option 1 reverse transcriptase. Next one. A vector that can survive in and be moved between more than one types of host cell is option 1 T-tailed vector, 2 shuttle vector, 3 moving vector, 4 labeled vector. Answer is option 2 shuttle vector. Next one. An area of microbiology that is concerned with the occurrence of disease in human population is option 1 immunology, 2 parasitology, 3 epidemiology, 4 bioremediation. Answer is option 3 epidemiology. Next one. Which process involves the deliberate alteration of an organism's genetic material? Option 1 bioremediation, 2 biotechnology, 3 decomposition, 4 recombinant DNA. Answer is option 4 recombinant. Next one. Which is the correct order of the taxonomic categories going from more specific to most general? Option 1 domain, kingdom, phylum, class, order, family, genus, species. Option 2 division, domain, kingdom, class, family, genus, species. Option 3 species, genus, family, order, class, phylum, kingdom, domain. Option 4 species, family, class, order, phylum, kingdom. Answer is option 1 domain, kingdom, phylum, class, order, family, genus, species. Next one. Family of proteins involved in second messenger cascades. Option 1 G protein, 2 P protein, 3 M protein, 4 T protein. Answer is option 1 G protein. DNA sequences found within genes that are involved in the regulation of patterns of development, that is morphogenesis, in animals, fungi and plants are called option 1 homeobox, 2 heterobox. Answer is option 1 homeobox. Next one. DNA methylation involves option 1 deletion of the methyl groups, 2 addition of methyl groups, 3 neither deletion nor addition, 4 addition of ethyl groups. Answer is option 2 addition of methyl groups. RI technology refers to option 1 RNA interfectants, 2 RNA interchange, 3 RNA interference, 4 RNA interleucinescence. Answer is option 3 RNA interference. Next one. Small infectious nucleic acid that does not have a protein coat is option 1 celluloid, 2 viroid, 3 vivid, 4 mucoid. Answer is option 2 viroid. Next one. Enzyme that changes the topology of DNA by nicking and resealing one strand of DNA is option 1 topoisomerase 1, option 2 topoisomerase 2, option 3 topoisomerase 3, 4 option 4 topoisomerase 4. Answer is option 1 topoisomerase 1. Movement of a transposon to a new site in the genome is called transposition, 2 transduction, 3 transcription, 4. Translation. Answer is option 1. Transposition. Next one. Receptor that moves small molecule across the plasma membrane is called option 1. Transfactor, 2. Transporter, 3. Transvector, 4. Translator. Answer is option 2. Transporter. Next one. Complex responsible transporting proteins from the intermembrane space into the interior of the organelle is option 1. TIM complex, option 2. TIC complex, 3. TAM complex, 4. TRI complex. Answer is option 2. TIC complex. Next one. Which episome of the bacterium Agrobacterium tumefaciens that carries the genes responsible for the induction of crown gall disease in infected plants? Example 1. Golgi complex, 2. TI plasmid, 3. RI plasmid, 4. PI plasmid. Answer is option 2. TI plasmid. Next one. Subunit of transcription factor TF2D that binds to DNA. Option 1. Tata folding protein, 2. Tata releasing protein, 3. Tata binding protein, and 4. Tata unwinding protein. Answer is option 3. Tata binding protein. Next one. Relationship between chromosomal regions of different species where homologous genes occur in the same order is called option 1. Monoctomy, 2. Synteny, 3. Parasynteny, and 4. Synonym. Answer is option 2. Synteny. Next one. Which DNA describes sequences that do not contribute to the genotype of the organism? Option 1. Replicating DNA, 2. Genomic DNA, 3. Amplified DNA, 4. Selfish DNA. Answer is option 4. Selfish. Next one. RTK is an abbreviation for option 1. Receptor tyrosine kinase, 2. Receptor trypsin kinase, 3. Receptor tyrosine kinetase, 4. Receptor trypsin kinetic. Answer is option 1. Receptor tyrosine kinase. Next one. Enzyme that functions in tRNA splicing is option 1. RNA ligase, 2. RNA galactase, 3. RNA lactase, 4. RNA leucase. Answer is option 1. RNA ligase. RNA virus with the ability to convert its sequence into DNA by reverse transcription is in option 1. Tobacco mosaic virus, option 2. retrovirus, option 3. ring spot virus, option 4. provirus. Answer is option 2. retrovirus. Next one. Short stretches of 1000 to 2000 bases produced during discontinuous replication of option 1. exons, 2. transposons, 3. introns, 4. Okazaki fragments. Answer is option 4. Okazaki fragments. Next one. Which enzyme is responsible for site-specific recombination? Option 1. integrase, 2. intergrase, 3. innase, 4. invertase. Answer is option 1. integrase. Next one. A genetic unit that codes for the amino acid sequence of a complete polypeptide chain is closely related to option 1. an anticodon, 2. a promoter, 3. a gene, 4. a codon. Answer is option 3. a gene. Next one. Given the antisense strand DNA codon 3' TAC 5', the anticodon that pairs with the corresponding mRNA codon could be option 1. 3' C5, option 2. 5' TAC 3', option 3. 3' UAC 5', option 5. 5' TAC 3'. Answer is option 3. 3' UAC 5'. Next one. Which of the following is not a characteristic of cellular RNA? Option 1. contains uracil. Option 2 is single stranded. Option 3 is much shorter than DNA. option 4 serves as template for its own synthesis. Answer is option 4 serves as template for its own synthesis. Next one. A mutation in the codon UCG to UAG is be described as option 1 missense mutation, 2 neutral mutation, 3 nonsense mutation, 4 frameshift mutation. Answer is option 3 nonsense mutation. Next one. An amino acid that cannot participate in alpha helical formation is option 1 proline, 2 histidine, 3 phenylalanine, 4 threonine. Answer is option 1 proline. Next one. A coding system in which each word may be coded by a variety of symbols or groups of letters is said to be option 1 archaic, option 2 redundant, 3 degenerate, 4 polysyllabic. Answer is polysyllabic, option 4. An amino acyl synthetase is responsible for option 1 formation of peptide bond, 2 attaching an amino group to an organic group, 3 movement of tRNA molecule from A to P site on a ribosome, 4 joining an amino acid to a tRNA. Answer is option 4 joining an amino acid to a tRNA. Next one. The step in the flow of genetic information wherein DNA is copied into mRNA is called option 1 translation, 2 transformation, 3 replication, 4 transcription. Answer is option 4 transcription. The growing polypeptide chain is first attached to a tRNA molecule in which site of the ribosome? Option 1 A site, 2 B site, 3 P site, 4 active site. Answer is option 3 P site. Next one. First protein sequenced is option 1 inulin, 2 insulin, 3 immunoglobulin, 4 albumin. Answer is option 2 insulin. Next one. First enzyme extracted in crystalline form. Option 1 invertase, 2 maltase, 3 urease, 4 amylase. Answer is option 3 urease. Next one. Central dogma of molecular genetics was proposed by option 1 Watson, 2 Crick, 3 Morgan, 4 Pasteur. Answer is option 2 Crick. Km value of enzyme is option 1 enzyme concentration, 2 substrate concentration, 3 rate of reaction, 4 activation time. Answer is option 2 substrate concentration. Next one. Human chromosome number was first given by McClung, option 1, 2 Berner and Home, 3 T. Geo and Levan, and 4 Crick. Answer is option 3 Geo and Levan. Next one. DNA in bacteria is a option 1 circular double-stranded DNA, 3 circular single-stranded DNA, 3 linear double-stranded DNA, 4 linear single-stranded DNA. Answer is option 1 circular double-stranded DNA. The genetic material of prions is option 1 RNA, 2 DNA, 3 lipids, 4 proteins. Answer is option. Next one. The 2 polynucleotide chains of DNA are linked by option 1 hydrogen bond, 2 phosphodiester bond, 3 peptide bond, 4 glycoside bond. Answer is option 1 hydrogen bond. Next one. Genes for RNA synthesis is present in option 1 nucleolus, 3 telomere, 3 centromere, 4 pellicle. Answer is option 1 nucleolus. Non-epistatic interaction was given by option 1 Morgan, 2 Sturtevant, 4 Bateson and Punnett, 4 Sutton and Bowring. Answer is option 3 Bateson and Punnett. Next one. Linkage can be detected by option 1 only test cross, 2 only F2 data, 3 both test cross and F2 data, 4 reciprocal cross. Answer is option 3 both test cross. Next one. Which of the following is not characteristic of most mRNA processing in eukaryotes? Option 1, addition of a poly A tail at 3' end. Option 2, addition of an unusual guanine at the 5' end. Option 3, removal of leader and trailer sequences. Option 4, deletion of introns. Answer is option 3, removal of leader and trailer sequences. Next one, transduction was given by option 1, Beadle and Tatum, 2, Cinder and Lederberg, 3, Lederberg and Tatum, 4, Avery, MacLeod and McCarthy. Answer is option 2, Cinder and Lederberg. Next one, 1% recombination frequency is equal to 1 map unit, option 2, 10 map units, option 3, 0.5 map unit, 4, 100 map units. Answer is option 1, 1 map unit. The size of muon in Benzer fine structure of gene is option 1, 1 base pair, 2, 10 base pair, 3, 100 base pair and 4,000 base pair. Answer is option 1, 1 base